So first of all, everyone, thank you for coming and thank you for staying through the difficulties we just had. My name is Casey Galvin, and I am a research engineer at Elix. I'm going to tell you about Elix Discovery, which is an AI drug discovery platform that we're building at Elix. But before I tell you about our platform, let me give a brief introduction for Elix itself. We're based in Tokyo, and in the past about 5 years, we've had a chance to work with a number of different corporate partners, some of whom you can see listed here on the slide. We've also enjoyed working with academic partners, both within Japan as well as international collaborators, and have had the pleasure to participate in 2 programs, one hosted by Blockbuster Tokyo, another through the NVIDIA Inception program. And through all of these engagements we've had with our partners, we've really developed an internal expertise related to artificial intelligence and molecular systems. And as part of that, have built internal tools to help us address the challenges and meet the opportunities that are available in this field right now. And so it's our goal with Elix Discovery to take the tools that we use inside of Elix and put them into the hands of chemists so that they can use them inside of the lab. Now, to do that, we've really packaged 3 things into the Elix Discovery platform. The first, which I just mentioned, is our experience and our intuition. The second is a set of state-of-the-art models that we have implemented based on their publication in the literature, and that we keep up to date and continue to update as new models and architectures appear. And the third part of Elix Discovery is that it's both modular and extensible. And I'll describe this in the next slides, exactly what all 3 of these points mean. But the key to moving it from our own resources inside of Elix out into the world, and especially into the hands of people who are not technical or machine learning experts, is to have a very intuitive user interface. And we've designed this user interface really with 2 kinds of users in mind. So the first person that we imagine interacting with Elix Discovery is actually someone like ourselves, someone who is perhaps a computational chemist, maybe actually a machine learning engineer, who works in a pharmaceutical company, is comfortable with the tools that are used in machine learning, can write code, but doesn't necessarily have a platform, and would benefit from having an integrated single place from which to build tools, implement custom models that could then be used throughout the entire company, or at the very least, the rest of the users with access to Elix Discovery. And that brings us to our main target, our main user that we have in mind, which is someone like a medicinal chemist, someone who is a non-expert in machine learning, someone who really works in the lab and doesn't write code. And our goal here is not to replace this person, but rather to provide tools that help this non-expert really take advantage of deep learning. And so there's a feedback here where Elix Discovery is able to provide predictions, it can do generative models, but at the same time, the chemist is able to put in his or her knowledge using some of the tools that we've implemented within the Elix Discovery platform. And the ultimate goal, of course, is to have data-driven decisions once it's time to start moving molecules down a pipeline towards a pharmaceutical product. Now, I'll tell you more about Elix Discovery, and let's start with this idea of a modular, extensible architecture. On this slide is a figure that shows an overview of the Elix Discovery architecture. And you can see here a number of different modules. we see the Elix process module, which is our set of tools for both bringing in data from different sources and applying standard pre-processing techniques that prepare the data to be used in different machine learning applications. We also have Elix Predict, Elix Create, and a series of other modules here. And these are really the heart of the Elix Discovery platform. Elix Predict contains all of our predictive models and is really the foundation. From here, everything flows. Within Elix Predict, we offer both graph models, which are really the state of the art within molecular sciences, as well as classic machine learning models, MLP architectures, because these are sometimes actually work just fine for the tasks, but also provide an important baseline. Elix Create is our generative package in which we've implemented a number of different generative architectures that work differently, that work better in some situations than others, and importantly, can incorporate models from Elix Predict directly, because they're both integrated on the same platform. So in this way, you can use Elix Predict to drive Elix Create to generate molecules that have tailored properties. Two other modules that we are currently developing include Elix Assist, which provides an active learning framework, which also incorporates Elix Predict in the way that it works, as well as Elix Synthesize, which contains retrosynthetic models that we've developed within Elix. Now, you can see this modular architecture would allow a customer, a user of Elix Discovery to really fine-tune what's necessary for their company goals. While Elix Predict is really a necessary component, not everyone necessarily does generative models. And so there's no need to include Elix Create if that's not your goal with the platform. Similarly, you don't necessarily need to include a retrosynthetic module, but you could certainly include Elix Create. So it can really be tailored to the specific needs of the company. Finally, to touch on the idea of the extensible architecture, we note down here that you could have custom modules, as well as really this idea of other. It's whatever you can do with it. And part of that is building in an API within Elix Discovery that allows companies, developers inside of companies, to access the tools within Elix Discovery and make sure that Elix Discovery can fit into your company's pipeline, into their workflow. Because once again, our goal is not to replace, but really to augment what is available. The final module that is perhaps just as important as all the machine learning is the ability to really understand the data that results. And that's done in Elix Wizard, which is our abbreviation for Visual Wizard. And so from here, this is where you're able to filter data, you can color code data, you have access to explainability techniques within AI, as well as a large suite of plotting capabilities. And this is ultimately what provides the feedback, which can lead to those data-driven decisions. Now, in the next slides, I'll show some screenshots from our current user interface to give you an idea of what this looks like. And our goal is to really have the complexity of setting up a machine learning, especially deep learning model. The default behavior that we would like to achieve is that a non-expert in machine learning can come to Elix Discovery, simple point and click, no code environment, and use nothing but the default values and still have reasonably good results as a starting point, and not be fooling themselves into thinking this is somehow better than it is. And so to give an idea of what this looks like, we see here how one would set up the creation of a new predictive model where through a drop-down menu, you can select an architecture. You can see here a set of graph neural network architectures, as well as classic machine learning models. Now, this would be for a regression classification task. You have the same access to regression models down here. And from here, clicking continue would lead to a series of steps that would guide the user through setting up hyperparameter optimization, which is ultimately done automatically, going through the training process, and really getting everything started based on the intuition we've developed and the way we would approach this, so that the user starts down the right path and ends up, hopefully, exactly where they want to be. Now, I mentioned before the automated hyperparameter optimization capabilities. This is, of course, a critical piece of developing good performing models. Rarely will you get the best performance from your initial starting conditions. And at the same time, it's probably one of the trickier concepts to understand from the beginning. So we've really automated the whole process. We actually use this internally as well, because we wanted to make it easier for us. And the key, one of the key usability features is that after going through all of the optimization, the results are presented in an easy to understand format that lets any user of Elix Discovery quickly identify which model to move forward with. And so this screenshot gives you an idea of this, where we can see the metrics for this particular run. This would be a classification task. So we have the different metrics plotted as a function of, plotted as a function of the different runs. And so you can see, okay, this is how models performed over time. Here you have a table from which you can identify the best performing checkpoints. You're able to sort according to the different metrics and choose different models based on that. And then I think to really understand, well, which one is working best for my task, because this is a classification task, we have a confusion matrix, which really just at a glance lets you see, well, are we getting good false, like accurate results in terms of identifying false values, as well as getting true positives, and likewise the case for false negatives and false positives. And so from here, without really having to understand how one would go through and analyze all this data, we present it for the user to make a decision to move forward. Now, once you have your model, that's great. You can do some predictions on your data sets. You can start to get some results and start thinking about, well, what are the best molecules for me to use, and start to choose a lead candidate from this. This is our envisioned use case. And to do this quickly, it really helps if just by looking, users are able to understand, does a molecule pass the filters that I have set in place. And so as part of that, we've created this no-code analysis package where a user is able to apply filters to filter out data that is in certain ranges, but also can apply color filters. And so these red and blue and green and yellow squares that you see are really applied based on the values that are in those squares. And so from this, you can quickly say, well, okay, anything that's red, I don't want. Okay, this is a promising molecule perhaps. And oh, here, this is green, that's great. It makes it easy for non-technical users to approach and quickly find the answers that they need from the data. Let's see. Okay, it did play, great. Now, like I said before, we don't want to really disrupt common workflows for chemists. And coming from a chemistry background myself, one of the things I often did was draw molecules. And so we thought it would be great if it would be possible to combine the ability to use something like a molecular drawing tool, like you see here, along with the models from Elix Predict, and ultimately make a real-time prediction based on the molecule we just draw it in this kind of tool. And so that's what you see here. So the video shows that the molecule is being put together with the chemistry that we want to investigate. After cleaning it up, we click submit, and we see that the molecule is now added. Oops, sorry, let me take a step back to this table. And along with that, the predictions were made regarding these two values, which were part of the predictive model that was trained previously on this data. And so in this way, the user, without having to set up some complicated spreadsheet with all the smile strings and really think about how to set something up for a machine learning model, can simply start from, say, a common scaffold that's already been identified, introduce the changes in the chemistry that they're interested in understanding, click submit, and quickly gets feedback right away on whether those changes in the chemistry are moving in the right direction, or whether that's maybe a direction we don't want to go. And so we think this tool is very cool. Certainly I do, and is really, again, the kind of thing we have in mind for our typical user, who is not a power user and coding up all their own modules, but instead someone who really understands chemistry, but still wants to use deep learning. Oops, you're going to play again. Okay. All right. The final module that I want to give an overview of that we're including in Elix Discovery, certainly not all of them, but the last one I'll describe is our active learning module, which we call Elix Assist, because it assists you with planning your experiments. With active learning, we have found this to be quite useful in cases where clients and our partners have small data sets. Now, data is really required to make a performant machine learning model, and sometimes that data just isn't available, especially in pharmaceutical sciences, where it's, one, expensive to do this, and sometimes because of patient confidentiality, you simply don't have access to that data. So in the case where a user is committed to building up a data set for machine learning, or at the very least is interested in trying to optimize the series and sequence of experiments that are performed, this is where active learning can come into play. And this is integrated directly into the Elix Discovery platform, along with Elix Predict. And that's why you can do this active learning loop quite easily with very little setup. And the way this works is you might start with an initial data set, and you'll train a machine learning model on this. It's probably not going to be the best performing model that's ever been published, because our assumption here is the data set is small. But nonetheless, you can then make predictions on a larger set of molecules that you have. Maybe your company has a library of molecules available, and you're interested in seeing if there are any promising candidates out of that. From these predictions, the Elix Assist module is able to identify top candidates according to a choosing algorithm. And these are ultimately the ones that will be performed or be used in subsequent experiments. So instead of looking at all of the molecules all at once, you're really choosing the ones that, according to this framework, are the most promising to move towards the best possible molecule out of the data set. And so this process then repeats, where this new data is added into your initial data set, you retrain the model, you make the predictions, you choose the next set of candidates, and collect that next set of experimental data. And in so doing, the hope is that you reduce and ultimately minimize the amount of experimental resources and time spent collecting that data before finally you reach a point that you can say, oh, actually, these candidates meet our requirements. So in such a data scarce scenario, which we often find in chemical sciences, we think this can add real value within an actual working chemical pipeline. All right, so as just a brief summary of the different modules that we plan to roll out the Elix Discovery platform with, we have Elix Predict, which provides the deep learning and classic machine learning models for property predictions. Elix Create provides a set of generative models. Elix Assist, I just described, is our active learning framework. I didn't describe Elix Synthesize, but this provides retrosynthetic capabilities. And then finally, Elix Wizard provides visualization for that data. And of course, anything you're able to produce, or if you like to work with us to help produce it in development, is also available on Elix Discovery. Now, I'll show you the way you can get in touch on the next slide. But to give you an idea of how this would proceed, once you contact us, there will be some kind of initial consultation where we really get to understand what the company goals are and any specific project requirements. From there, we can start to tailor Elix Discovery, including the module selection, as well as any custom development that might be necessary if there's specific technology within your company that would require a custom module. That's certainly something we can do. Finally, we would then deploy Elix Discovery, and this is something we'd probably work with your company IT department on. We have experience doing this. We're using company resources, and usually the IT department is quite concerned about how we go about doing that. But it's something we're happy to work with our customers with. And then finally, because this is an extensible architecture, we can provide ongoing support in the form of updating the models that are available as new models appear in the architecture and new machine learning techniques appear as well. So with that, I'd like to thank you for your attention. Arigatou gozaimasu. And if you are interested in learning more, you can visit our website or reach out to us at info@elix-inc.com. Thank you very much.
Thank you, Casey. ご質問ございましたら、お願いします。いかがでしょうか。ご質問、もしメールアドレス書いております通り、info@elix-inc.com にご連絡いただければと思います。もしご質問がないようでしたら、次の演題に進ませていただこうと思います。続きましては、成果であります、ターゲットディスカバリーin the age of big data and AI ということで、ナジムから、発表を、行わせていただこうと思います。ナジム、can you hear me?
Yes, I do. Can you hear me?
Okay, so please start.
Okay. Are you able to see my screen?
Okay.
Yeah, thank you. Let me move to a broader one. Hello and welcome everyone to this presentation. And in addition to what my colleague Casey has explained and introduced our Elix Discovery platform, at the company we value internal R&D research and taking grand challenges in revolutionizing and assisting major problems throughout the pharmaceutical drug discovery pipelines. And today, I would like to introduce you one topic that we are already also interested in, which presumes a grand challenge overall in the pipeline, and try to introduce you to the problem setting and potential solutions. So this is more of a hypothetical presentation, which includes a topic that is of value to both industry and ourselves. So I would like to first start with the overview. I would provide a brief background and the components of the problems. I'll touch upon potential data sources, which is a subcomponent in the problem components, and potential approaches briefly. And throughout the talk, I'll touch upon some challenges and opportunities in the field and conclude the presentation, and hopefully transition to the Q&A. So as a background, throughout the drug discovery pipeline, selection and prioritization of novel drug targets is a central problem, because choice of the right target can often significantly impact the probability of advancing of the drug to clinical development and also affect clinical trial success. However, this is not an easy problem, and that is one of the reasons we are also interested in this problem, because in this type of problem, you have to optimize a large variety of parameters that are interconnected when you think of it as from the biological perspective. So considering identifying a target, some of the things that are really important is link between this target and the disease. What is the target role throughout the disease progression? Whether this target has novelty in terms of like if it's how much information is about this target regarding its involvement in the disease, if it is of interest to pharmaceutical industry to proceed and choose this target and to go all the way through the clinical trials. Initially, of course, how druggable the target itself is, considering small molecule modality. But our thinking is not only limited to small molecules. And of course, the safety and the list goes on, basically. That's why this is a type of problem that we think is applicable for machine learning setting, given that some prerequisites are met. And if we consider the challenges, so far the challenge in the industry is that since human genome has been more or less understood to some extent, we learned that there are around 20,000 proteins expressed in human genome, and it counts only to a small fraction of the human genome. And out of those proteins, we know that around up to 15% of those at this moment in time are considered to be disease related, which is also due to the lack of ongoing research in this field. And out of that fraction of the proteins, so far we were able, like in the industry as a whole, was able to address around less than 700 of these proteins to target them and with different modalities. However, a majority of disease-related protein targets remain undruggable, which means either they, so mostly they lack distinct binding motifs pockets for small molecules to bind, especially when small molecule modality is of concern. And another problem and another challenge is a question more of a like theoretical question, considering if it's only like focusing on the protein targets only, if that is the best way to go, or obviously it has proven a certain level of success, but considering the targets, especially when we consider undruggable targets, considering the protein itself per se might not be the most effective way to go, and there might be additional options. So then the question that I'm trying to present and propose in this talk is how to identify novel targets, considering this complexity of the system. And subsequent question that follows is, can AI be used to facilitate this process as a whole? But to be able to address these questions, I'd like to take a look at what we need, considering application of like techniques like machine learning to such a problem. Obviously, we rely, so the limiting factor in this setting is data, and understanding what type of data is available, what's the data structure, how to represent this data to be compatible with methodologies and what type of methodology to choose, what type of architecture to choose, how to optimize it, etc. And let's start with overviewing the data. So fortunately, for us as a community, recent advances in sequencing technologies and many other technologies have led to robust and exponential accumulation of data sources, which are deposited and accessible to the public. And we have a lot. These are just a very small list, but we have very large-scale human genomics, proteomics, and metabolomics data sets that come, and they are continuously accumulated. We have some useful resources like UniProt, and where a lot of information on the protein targets, and not only targets, on the proteins as a whole is accumulated. And we have therapeutic targets. A lot of effort has been actually put into establishing these data sets, and they grow exponentially. And they grow to a degree at which, for humans experts, it becomes less and less possible to keep track of all of the data, and it makes it harder to analyze and make use of all these valuable data. It would be nice if, of course, combining all this information in a single place for further analysis. However, this is a very daunting task, considering the different nature of all those data sets and different information that is inherently saved in each one of those data sets. Because they specialize in different areas, some of them on protein expression profiles, on disease association, pharmacology, gene expression, protein, like gene-gene interaction networks, protein interaction networks, and the list goes on. And navigating through a myriad of cross-references in such a huge space in order to paint an accurate portrait of a potential target is necessary, but at the same time, it's a very challenging task. And this might be one of the contributing factors to the low success rates that we see in the clinic, because quite often we lack general understanding, general knowledge of involvement and pathways, all the interactions that the target that we chose is involved with, and quite often some of the important side effects and inefficacies only come up very late during the process. So having a system that would actually be capable of navigating good enough to a certain extent in this large space of biological network data would be an interesting thing if we can really leverage this all this data and to our benefit, and be able to draw useful information related to the different targets that might contribute to the disease of interest and many other underlying biological processes. So, but in order to use all this data, especially in machine learning setting, we need to find a way to structure all this data and to be able for the algorithms to utilize this very unstructured and data. But when we take a closer look at it, especially at the biological networks, we can see that be it protein interaction networks, gene interaction networks, metabolic networks, and many other networks, this type of data actually can be represented as graphs that capture interactions between nodes, and this type of graph, like graph structured data, is already being used, and graph-based algorithms have already been successful, especially with small molecule domain. And this is, I personally see as advantage. Given the complexity of the system, being able to represent it as a graph and potentially utilizing it, this whole huge amount of data, and use graph theory and leverage it to understand the relationships between the nodes in this huge amount of data should be an interesting approach, and might be useful in obtaining the hidden features, hidden patterns that we often, as human experts, fail to retrieve given the complexity of the system. And of course, machine learning techniques, which is the main focus of our company. We think that they can be helpful in processing this type of data and identifying features related to the successful target selection. Of course, this does not mean that we undermine the complexity and underestimate the complexity of the system. Rather that this is a challenging task that needs a solution, and we are willing to tackle the important problems that will have direct contribution to the field. And this is one of the things in the coming to deep learning. So one of the strengths that we see with deep learning is that it provides, it sort of helps us gives us more ability to detect complex pattern in data. And actually this particular property makes it quite suitable for the application of bioinformatics and this type of the problems which involve complex data and biological network data, and particularly usage of this information is relevant to early and de novo target identification. And the fact that in the recent years, deep learning methods have been extended to graph structured data makes it a promising technology to tackle these biological network analysis problems. Given said that, so through the overview of the literature, given the problem setting of target identification, most of the studies so far have focused on more or less repurposing approaches where people try to solve the drug-target interaction problem, where there are drugs that are attempted to be linked to known targets, and vice versa. However, in this particular talk, I'm actually emphasizing the other part of it, which is a more grand challenge, where the main idea is to utilize these technological advances in deep learning to actually suggest totally novel targets, identify novel key players in the disease, novel pathways from this biological network data. And so this is the field that has been just only recently gaining attention, and even if due to the appealingness, like attractiveness of the problem, there are very few studies related to that. So basically, the field is very under development and in infancy, which strikes me personally, because the accumulation of this much data actually should be, in my opinion, a facilitator. But of course, the complexity of the system most probably is the main reason why still there is a huge gap in this potential field. And so thinking of what type of things actually we can predict, what type of things we can do. So this is just a simple proposal. So one thing is that once we can establish and organize those different data sources and link them between each other, then it should give us the ability to navigate through the interactions between different genes, diseases, targets, drugs, and all that interaction that can be that is already available in the publicly available data, and predicting unknown functions of certain nodes. And this can be involvement of a gene and role of that particular gene in the network, how central the role is, what type of pathways and other pathways it intercepts with, and the significance of that particular node in the other networks. So this type of data and is crucial because when we target a certain protein, or let's say a node in the system, understanding how targeting this particular node will affect the other underlying systems actually is one of the keys to being able to estimate a potential off-target and side effects that can come up by targeting this particular node. We can attempt to predict the interactions between different nodes. So the associations between certain diseases, genes, and proteins, their interaction between each other. So what type of interaction is going on, which is a bit difficult thing to man, but still with the accumulation of this data and further structuring, this type of things that have the potential to be achieved, and once it is done, it can be a big contribution to the field, and it can facilitate especially and target the especially very early stages of drug discovery, which is target identification. So with this, I'd like to have some concluding remarks because the aim of this talk was more hypothetical and thought-provoking, and to present an area of the field that we personally see understudied, and a huge gap which has huge opportunities, but at the same time being challenging. So novel target identification remains a challenge, but yet the centrality of this problem makes it at the same time attractive. And obviously the involvement of myriad of parameters contributes to this problem complexity. And there is a huge need of robust ways to process this large biological network data and suggest novel target candidates, and try to solve mechanisms, underlying mechanisms, even in the diseases that are currently being addressed, not even mentioning the diseases that we weren't able to attack yet. And as an advantage, so the complexity of this system can be more or less structured into a more usable format, which is graph format, and can be subsequently analyzed using recently developed models, especially graph-based models. And so far to this stage from the literature survey that at least we've been doing, it seems that most effort has been focused on more drug repurposing related tasks like drug target interaction prediction problem, which is also interesting and an important problem to be solved. However, this major field still remains unaddressed to the large extent, which makes it quite attractive and important field. With this, I'd like to thank you and conclude my presentation. And if there are any other, if there are any questions, I would be happy to discuss them.
Thank you, Nazim. ご質問ございましたら、お願いします。いかがでしょうか。ご質問がもし、ございませんでしたら、次の、演題に移らさせていただこうと思いますが、よろしいですかね。大丈夫でしょうかね。 はい、そうしましたら、続きまして、弊社の、研究者でありますジュンジンから、towards generating synthesizable de novo small molecules ということで、次の演題に移らせていただこうと思います。ジュンジン、can you hear me? Okay.
Hello, can you hear
Okay,
me?
Junjin. Yeah, okay.
All right. Okay.
Okay, please start.
Okay. Good afternoon, everyone. My name is Jun Jin, and today I'd like to talk a bit about generating synthesizable molecules, especially from a machine learning perspective. There have been recent trends on generative models for generating new molecules, but most of them were trying to achieve something like getting novel new molecules instead of beating the bench, instead of trying to produce a real world experience of trying to simulate the idea of generating a novel molecule that is synthesizable. So with that, today's topic, I'd like to touch upon several things. For the first part of this talk, I'll briefly talk about generative models and what defines a generative model. And following that, I'll talk about the paradigm that is presently being used by generative models to induce this synthesizability a property into the generation of the molecules. And finally, I'd like to touch upon the pros and cons of each approach, especially from the perspective of a synthesizable molecule versus a novel base model approach, which brings us into two other models. And with that, I will conclude with this talk. So before I begin, as some of you may know by now, a generative model from a machine learning perspective is basically a model that generates something. But from a probabilistic approach, we can define the idea of generating a molecule as trying to map a input. For instance, if this is the representation of a molecule, we can first map it into a computer comprehensible input like a graph or a small strings, for instance, and we would try to map it into a low dimensional space. And this space here is usually called the latent space. And what we are interested in this latent space is the landscape of this latent space, so that's in such a way we can sample from it. In most cases, we will try to fit a model in this latent space using any model that we can think of, but from a probabilistic perspective, we can use a Gaussian distribution or even delta distribution or any types of distribution. The idea is as long as we can fit a model in this space, we can start to sample from it. Once we sample from it, we would put it into a decoder, which would then decode this latent space information back into our real information space such that we can obtain the true molecule that we're talking about. In most cases, this is defined in a mathematical form where we are trying to learn the joint probability of the data, which is x here, marked by x, and the latent variable z. So if we can ideally learn this entire space, we can basically sample any type of valid molecule that would then be possible to generate new molecules. But in most cases, this distribution here is intractable. What this means here is that if we try to compute it or try to perform some kind of computation, it would be taking forever or ages to basically be able to learn what this distribution is. So in most cases, we try to approximate this distribution through some kind of heuristics or through some kind of proxy function that would anticipate what this true distribution is. For instance, one of the most popular model in the generative perspective is called the variational autoencoder. And similarly, this architecture that I've drawn up here simulates the experience of a variational autoencoder. What the input here is given here is a small string. It can be a graph or it can be just a text form in terms of small strings. And we will insert it into the encoder, which is a machine learning model. For instance, it could be a neural network, or it can be any type of model that would bring it down into a latent space. And what we are interested in is we would like to map out this latent space using some kind of latent variable marked by Z, and we can then try to interpolate within the space so that once we sample from this distribution, we can obtain back the true molecule. Ideally, if this space is mapped well, we will be able to obtain all kinds of different molecules in terms of like novelty, synthesizability, and even the properties that we'd like to ensure. This is the ideal case, but in a real world situation, this is very difficult to be done. Sometimes there are issues like the distribution that we have chosen may not be suitable. So from the start, we are setting on the wrong foot. So we never know what this true distribution is and what kind of a proxy distribution that is suitable to solve our problem. So this brings us to the generation paradigm. Imagine that you have a generative models that can generate a molecule. How would you enforce the generative model to introduce some kind of synthesizable constraint? In a general perspective, from a traditional point of view, trying to come up with the novel molecule, we would basically start with a filter of a database that consists of all kinds of synthesizable molecules. From there, we would filter out molecules that are not synthesizable or molecules that are not within our property framework and so forth. Once we have taken this database and filtered it out, we would put it through an evaluator. Our evaluator here is then a mechanism to ensure that whatever property that we're trying to optimize would be maximized here. For instance, if solubility is the main goal, then we need to ensure that whatever molecules that we have sampled from the database should satisfy this criteria. Finally, we'll use this as a starting molecule scaffold and try to fit to our target and see if it works. In most cases, a generative model would stop at this process. However, a recent trend with generative models is that we can be very flexible with fitting into this synthesizability constraint. For instance, the first approach here is just a standard approach without concern for the synthesizability constraint. So we basically replace this database with a generator that will help us pick the most likely molecule that we can use and try to combine it with a different scaffold or change its form so that we can come up with a new novel molecule. Once we have the new novel molecule, we go through the same process and evaluate its property. This is the most standard approach. In most cases, this approach here will not guarantee us some kind of synthesizable molecule. Hence, there are other approaches that we need to consider. One of the simplest methods here is to consider including a filter right after the generator. So once we have generated a new molecule, we evaluate the property, and if the property is what we want, we proceed to the next stage, and we ensure that this is a molecule that is synthesizable. Using some kind of heuristics or some kind of maybe oracle or even a real-world medicinal chemist, we ask that person if this molecule is synthesizable. Then we decide to pick this molecule as one of our starting points. And in most cases, if it's not synthesizable, we tell the generator this is not synthesizable, and the generator we will retrain from the database. This is the post hoc filter approach. The alternative here is to include some kind of bias into the database. So assume that the database we have here is it's a whole set of molecules that include molecules that are synthesizable and non-synthesizable. The simplest approach here is once we ensure the database here is always a synthesizable molecule, the pipeline towards this approach here would always ensure that we are telling the generator to only learn how to generate synthesizable molecule. This is one way. The next approach is to consider a heuristic. So in this C case here, we are using some kind of synthesizable scoring function or heuristic or even a medicinal chemist. But in real world, trying to use this kind of medicinal chemist is very costly. Each time we probe a medicinal chemist, we have to pay a sum of money, and that's very expensive. And this process could be very slow. So the alternative here is to design a heuristic function that we can approximate what the idea of synthesizable molecule is from a true medicinal chemist, for instance. So this heuristic here would then be able to tell the generator immediately if this is synthesizable or not. Another approach to considering a heuristic is an upgraded approach, that is to use a more advanced oracle. And this oracle here, it tends to be a very huge complex model that is trained on thousands of molecules or even billions of molecules. And they are capable of telling if this molecule is synthesizable or not. In some cases, this oracle is available. In some cases, it's not. Sometimes it's available through some kind of fee, and it's proprietary, so we never know. Finally, the most recent approach is to consider some kind of explicit constraint. This explicit constraint here is a little bit different from bias and all the other approaches. What we want to ensure here is that the constraint that we're enforcing in the generator is restricting the chemical space that we're trying to access from the start. So in this case, we are only making sure that the database here is synthesizable. What if we want to ensure that the molecule itself in the database is also purchasable, also a building block towards a more complex molecule, for instance? So this kind of explicit constraint starts to stack up as we start to add into the generator. What this here shows here are basically figures that I've taken from this paper, Gao here. You can find the reference later. But overall, the paradigm of trying to generate a synthesizable molecule can be summarized in this couple of steps. What we are concerned here is that trying to generate a reasonable synthesizable molecule and trying to pick the best approach here. So let's go through one by one. Among all the other generation paradigms that I would like to choose, one of them is called the bias by training. And as I mentioned before, bias by training is basically trying to constrain your database from the start, and we ensure that all our molecules within this database is synthesizable from the get-go. Imagine you have a molecular space like this from a blank canvas, and we are not sure what the contents are. We start with a constraint space of saying that this database has some kind of molecules, but they may be not synthesizable as well. If we constrain this further, we will achieve some kind of set of smaller subset here, which are synthesizable. We would then train our generator with this. This approach is has its advantages and its disadvantages. The advantages here is that we are starting with a set of synthesizable molecule from the start. So the model would start to learn something very quickly. But this would then give us some kind of leverage on the success rate when the generator starts to go through the evaluator. So we may not need so many number of training as we start to go through this process. It's also fast and easy because the number of steps that we need to train the model may be reduced tremendously. However, the disadvantage here is that once we have constrained the space of molecules that we're looking at, we are basically shrinking the number of training set we have on the database. This basically leads us to problems common in machine learning, like local optima problem. So whatever molecules that are generated by the generator may or may not be the most optimal one. And they may differ only by a slight change, which is not something that we are looking for if we are seeking for more novel molecules, which brings us to the point that this approach may be limited in terms of novelty. The next most popular approach would then be to use a heuristic. Since heuristics are fast and computationally effective, we can consider this approach as one of the most popular approach these days. How we can do this is basically by incorporating the synthesizability loss as a loss function in a machine learning model, for instance. If a typical machine learning model usually needs to compute its loss by some kind of way to backpropagate its signals to teach the generator. In an unsupervised learning model, most of the time we are concerned with the reconstruction loss. That is to say, the input has to match the output. And occasionally we will add a regularizer to ensure that the model doesn't try to memorize the database. So that's one point. And we can then further make this more complicated by including a synthesizable loss function. from the heuristic. The synthesizable marked in bold can simply be a binary choice. If the synthesizable heuristic says this is synthesizable, then we put it at 1, or otherwise it's at 0, and so forth. Or it can be some kind of percentage in a range of 0 and 1 to tell us how likely this loss is closing into a synthesizable molecule. So the advantage of this approach is that we are more flexible with the choice of models. And because most of the machine learning models would use some kind of loss like this, so a model like reinforcement learning would be most suitable to learn to use this kind of a heuristic function. One of the advantages here is that it's easy to implement. So since the choice of machine learning models can vastly vary, we can basically implement it very quickly. The difficulty here is that it becomes more difficult to train. As we start to include more constraint in the loss function, the space that we are looking into starts to shrink. So this is the opposite of what we really want to look into a novel molecule. Also, the heuristic may be very naive, depending on what kind of heuristics we use. And the next approach to the previous heuristic is to basically just replace the heuristic function with a more accurate oracle. The oracle is basically a proxy that would try to emulate whether this molecule is synthesizable or not. Usually it's through some kind of simulation or very, very computationally intensive approach. The advantage of this is that most of the time the oracle is already implemented, so trying to incorporate into our machine learning model is pretty straightforward. And it's also less likely to fall into our local optima. This disadvantage, though, is that usually this CAFS here is a closed source or it's proprietary, so we may not know the true answer to this. We have to assume that the oracle is always correct. In some cases, it's also very computationally expensive, considering if we need to train the oracle as well. And finally, the most recent approach is to consider some kind of explicit constraint from the get-go into the generative model. This approach here basically means that if we take back the molecular space that we have right here, we are just trying to pinpoint that this small dot here should include some kind of additional constraint we are concerned about, like purchasable compounds, or if they're widely available, and so forth. This approach here would help us in several ways, in that it's very useful to use this approach to generate multiple steps of a synthesizable compound. So in the previous approach, a lot of the generative models would only synthesize from the reactants to the product in a single step. For a more synthesizable approach, we are concerned with the steps that the generative model takes from step 0 to step Z, for instance. And a lot of this also helps in the sense that we can basically use compounds that are of public knowledge. So we are further constraining this idea of trying to use synthesizable compounds that are widely available. The disadvantage here, though, is that the search space becomes much smaller. So we may be restricted to compounds that are less novel. They are also much more expensive in some cases, depending on what type of synthesizable constraint that we need to include, because once we include more constraints, the number of training steps needs to increase. So there are also more dependencies depending on the where we obtain this particular data set. So the general takeaway here is that we can summarize this huge problem of paradigm into two parts. I would say I'll call this the novelty problem and also the exploitation, where the synthesizability problem. In the novelty part, we are concerned of discovering new molecules in a large chemical space. And in order to discover this, we no longer restrict our model to whether the model should generate a synthesizable molecule or not. This task is then passed over to another model called the retrosynthesis model. And that's the job of the retrosynthesis model, to identify the pathway from getting this new molecule from our purchasable compounds or our starting compounds. On the other hand, we have synthesizability built into the model from the get-go. And this ensures that the model is concerned of synthesizability from every step of its training. This is good in some ways. The idea of trying to match a real world experience of trying to find new compounds is more akin to the synthesizability approach. But the downside here is that we sometimes need to rely on a secondary models like a forward prediction model to predict the discovery of the next compound. So overall, there are some advantages and disadvantages, and how do we balance out this is still a research problem. And yeah, I would like to briefly touch on the challenges in the two models. In the novelty approach, we would consider a retrosynthesis model, and some of this retrosynthesis model are widely available, like the AI ZIS Finder, and these are very nice tools used to help us reverse search a particular compound. But most of the time, this types of model are limited to proprietary data sets like Reaxys, and the public has no access to. Also, this correctness of the data set may sometimes be inaccurate. As some of you may know, USPTO data sets do have problems. On the other hand, if we sway towards the synthesizability paradigm, we need to rely on a forward reaction prediction model. Like the retrosynthesis model, we need to train it using the same data set of USPTO or Reaxys, but oftentimes this is more approachable because sometimes we have templates to apply them, so we don't need to really rely on a more complex model, but a complex model always exists. So even though the two paradigms are very different, the end result of using a forward reaction or a retrosynthesis is still an open problem. There is no one good model to approach this. So in conclusion, synthesizable molecules are much more crucial for drug development, but they come with several caveats. They restrict the search space considerably, and this is very useful to teach the model, but on the long run, the model may start to fall into a local optima, and we may not obtain a very ideal new novel molecule. On the other hand, they rely on a secondary model called forward reaction predictive model, and sometimes this can be good and bad depending on the model. The advantage, though, is that since it simulates a real-world chemist experience, this is more realistic, and most of the time we can ensure that the starting compounds should be purchasable from the start. On the other hand, if we sway towards trying to achieve new novel molecules, then we have to remove the constraint. This is a good in certain sense, but there are also caveats. First, a lot of the models that rely on this approach are one-step generation approach. So we are basically taking the reactants and we get the end product. And in between, we do not know the steps how we obtain the end product. So we need to rely on a retrosynthesis model. This sometimes is much more computationally expensive than a forward predictive model. So that's also one of the caveats. The advantages are we are allowing more room for creativity, so we can explore the space as we see fit. This is very nice from a research perspective, but may not be so nice for a real-world chemist point of view. Some of the molecules may have better properties because we have basically allowed the space to be searched in a wider manner. Yeah, and another approach is basically, rather than taking all this computational expense, is we just design a much better heuristic function. So one of the couple of popular heuristic functions are SA score, SC score, and basically just a small string length. And I will not go through in very detail, but each of them have their own caveats depending on how we use them. And realistically speaking, if we have a function that could approximate what synthesizability is, then we can have a very nice model that would train much closer to the real-world experience. But of course, this coming up with a heuristic function is very difficult. So we often ask ourselves what really constitutes to the contribution of the synthetic complexity. We will never know unless we look into billions of compounds one by one and identify them. So that's what machine learning models are trying to do right now, simulate this experience of a synthetic complexity. Also, considering if it's capable of measuring synthetic complexity, how well does it consider other constraints? Like maybe some of them are easily purchasable, but very difficult to synthesize. Do we consider this as a do we consider this as a easy to synthesize compound, or do we consider it as a difficult compound? This is really subject to argument. So with that, this brings me to the end of my talk. If you have any questions, please feel free to ask. Thank you very much.